Good afternoon, everyone. We'll get started. Welcome to the April Food for Thought seminar, and just before the Easter long weekend, so I hope you've all got a lovely Easter planned. May I acknowledge the traditional owners of the land on which we meet today, the Whadjuk people of a proud Noongar nation, and we acknowledge their continuing contribution that they make to the life of our city and WA, and I pay my respects to elders past, present and emerging, and I extend that respect to all Aboriginal and Torres Strait Islander peoples. I'd like to remind attendees that the Foundation will shortly be accepting applications for our 2026 Career Advancement Fellowships Grant Program, which will launch shortly. And we encourage all of you to sign up to our Signet mailing list so that you can stay up to date with our grant programs. It is now my great pleasure to introduce our very own Strategic Research Advisor, Professor George Yeo. George received his doctorate in biochemistry from the University of Western Australia in 1972. Shortly after, he was awarded the C.J. Martin Overseas Research Fellow and studied at the University of Pennsylvania, USA, and the Beatson Institute for Cancer Research in Glasgow, Scotland. He filled many roles as visiting scientist at several prestigious universities in the USA and in Europe. And following his return to Australia, he was awarded an NHMRC fellowship and achieved successive renewals to the level of Principal Research Fellow. He was the Associate Dean Research in the UWA Faculty of Medicine, Dentistry and Health Sciences, and a Professor of Biochemistry and Molecular Biology, and is an Emeritus Professor of the University of Western Australia. He has an extensive protracted interest in liver progenitor stem cells, or LPSCs. Is that correct,
Yes.
George? LPSCs are induced in liver when rodents are placed on a diet, which results in fat accumulation that damages the liver. The Yeo Lab was also the first to demonstrate the presence of LPSCs in human liver diseases, namely chronic alcoholic liver disease, hemochromatosis, and hepatitis C. These findings suggest that LPSCs may play an important role in liver generation in liver pathologies, especially when damage is prolonged. Most of his publications in the last 10 years have focused on this topic, for which his laboratory has an established international reputation. His reputation is also founded on the quality of his publications, collaborations, and participation at meetings as a keynote speaker and reviewer for the leading hepatology journals. Today, George will be presenting on identifying anti-liver cancer drugs in marine sponges and establishing the utility of a biomarker to diagnose and monitor cystic fibrosis patients with a liver comorbidity. And he will speak on acquiring funding and progress reporting on outcomes. Please join me in making George welcome.
Thanks, Kate. I think I could even skip some of my introductory slides about this topic, but I'm very glad to be here. I think I did one of these Food for Thoughts 9 years ago, if I recall, and it's great to be back. So the title is based on what Daria said to me, George, you must speak on this, you must speak on that, and we want you also to tell us how you get grants. And so I said, okay, I'll do my best. But what has happened recently to me is really it's serendipity, because I've continued to do research, and I will show you what we are up to and how we got the funding for it. And hopefully you can use some of these tactics, grantsmanship, and win grants for yourself. All right, so let's progress this. I think I just clicked this, don't I? Aha, great. So the liver, over 500 functions, and I got into it because it's such an important organ, no liver, no life, and the liver has a considerable number of genes that it expresses that are very tightly regulated. because you realize that, in the neonatal, in fetus to adult, you've got newborns, all the diet is changing, and your body takes in all sorts of different compounds and chemicals, and the liver is the first port of call after it goes into your gut. So it is actually very, very important, especially in today's world, health-related lifestyle practices, excess calories, alcohol, infection, drugs, toxins, etc. The liver has to cope with it, okay? And it copes with it by making the right enzymes and the right amount of these enzymes in order to detoxify and metabolize. My study started when I was a PhD student, and we were dealing with rats at that time and doing a cesarean section on the fetal rat, simulating birth, and then looking at these liver enzymes. And my God, they change in level 100-fold within 12 hours after birth, from almost zero right up to very, very high levels. So the regulation of these genes was of interest to me. In fact, when I heard about all this, Professor Ivan Oliver was lecturing me, and I was a medical student, and I called my mother and said, I don't think I'm going to be a doctor, I'm going to be a researcher, Mum. And she was horrified. But that's what drove me into biochemistry and molecular biology. So this is a story that I adopted from my mentor, Howard Holzer, who was at the University of Pennsylvania. And I went to him because he viewed the development of organisms as stepwise progression, where cells in a stepwise fashion progressively acquire new properties by proliferating and differentiating. So to make us from a single cell, you've got many, many proliferative cycles to make more of you, but what I call differentiative cycles, which Howard named quantal cell cycles, with proliferation with differentiation. So on top of it, he was working on muscle, he was working on chondrocytes, and he was working on erythrocytes. I went there to learn how to culture the different types of cells, but to bring the technique home when I came back to Perth and apply it to the liver, which again is my favorite organ. So here, what I want to point out to you is that when the liver is damaged, hepatocytes very readily give rise to more hepatocytes. The liver is a very, very good regenerative organ. Why? Because it's got to cope with all those things that are out there that you ingest, and if it doesn't do that quantitatively, then your health is compromised. All right, so here, this is a different cell type of division. Here's a liver cell, which is a stem cell. L here is a liver stem cell, not a hepatocyte, but it can give rise to a hepatocyte. It can give rise to a stem cell. So this is an asymmetric cell division. Liver stem cell gives rise to liver stem cell and a hepatocyte. Liver stem cell gives rise to a liver stem cell and a chondrocyte. That's a cholangiocyte. So it can give bile duct as well as hepatocytes. All right, so these are what we call differentiative or quantal cell cycles, as Howard Holzer coined it. So in this particular instance, the stem cell gives rise to a cholangiocyte and a hepatocyte. But these are proliferative cell cycles. These are differentiative cell cycles. And that's what I really focused on in my first 10, 15 years of my research. And thereby, we then adopted the notion that the liver comprises of stem cells. It has stem cells, apart from the hepatocyte being able to divide. But when does it use these stem cells? Well, we found that if we stress the liver strongly enough and for long enough, we could induce these liver progenitor stem cells. And I'm just going to summarize some of our publications. So I focused on the liver stem cell, and I knew what a liver stem cell looked like because I knew what a fetal hepatocyte looked like from my PhD work. We then found that when we gave rats a lot of ethanol to drink, they were called oval cells in rats. The liver stem cell appeared. Okay. By the way, when I did this experiment, ethics got hold of me and said, "What are you doing to these mice?" Right? I said, "Well, come down and have a look." And there they were. They were all happy, rolling around, getting fat, and there was no problem with the ethics because the mice were happy, or the rats were happy. But they induced these stem cells. We also develop a high-fat diet that induces these oval cells in mouse liver. But what is interesting here that I want to emphasize is with this model, after between 3 and 6 months of fat-induced liver damage, these mice develop liver cancer. Okay? You might have heard in the news that people with metabolic-associated steatotic hepatitis go on and develop liver cancer. Okay? So here is the very beginning, so very early on, we had some evidence to say that high-fat diet induces and obesity causes liver cancer. Okay, to continue the story of looking at these cells in culture, I isolated cell lines from these mice that we've treated with the high-fat diet. And I went to the Pasteur in Paris because Mary Wise's lab announced that they could do this. I've been trying in Perth to get the cell line. I couldn't do it. I actually cheated. I used a mouse. There's another story, but I used a p53 knockout mouse and made a cell line. Published that. But then Mary Wise said, you know, we're smarter than George Yeo's lab. We can make them from normal mice. And that intrigued me. So I spent 6 months at the Pasteur, and Mary was good enough to pass on this secret technique so that I came back to Perth and I made liver progenitor cells from normal mice. The other thing I want to tell you about the liver progenitor stem cells is that we linked them with liver cancer. At the University of Washington in Seattle, I undertook these experiments where we showed that if they had a mouse with a new tumor necrosis factor receptor knockout, and we knew that this factor was responsible for making these liver progenitor cells appear, in this knockout mouse, they really did not produce the progenitor cells, and they suffered greatly. A lot of them actually died. And once again, when I started these experiments at the University of Washington in Seattle, I had to confront the animal ethics group who said that, look, George, you put them on the diet, and while the controls are able to make new hepatocytes from progenitor cells in the tumor necrosis factor receptor knockout mouse, that's blunted. And because they can't respond, these mice are actually dying. And I said, well, I'm sorry about the circumstance, but that does prove that this pathway of liver regeneration for the stem cells is essential for life. So that was an important outcome. So we also suggested that these cells can be Jekyll or Hyde and be good to fix the liver up, but there's a bad side to them, and that they could go awry and actually be responsible for liver tumors. And finally, what happens in rodents, mice, and rats happens in humans. With John Olnick, we published in the American Journal of Pathology this paper, and this has been cited nearly 700 times. We showed that these stem cells, liver stem cells, actually are present in humans who have alcoholic liver disease, chronic hepatitis infection, or genetic hemochromatosis, where the liver is stressed severely and for a long time. Okay, so we now have shown that these cells are very, very important. And the next lot of stuff that I did was I actually sent these cells because I thought that it would be excellent for cell therapy, right? Because we could actually rescue. Part of the experiments were, in fact, to hepatectomize mice and then give them these cells and see whether we can actually remove more of the liver or make the mice really sick, but rescue them if we implant these cells into them. And friends at St. Vincent's in Melbourne, they were doing a lot of these kinds of experiments, and I sent my cells to them. Kiryu Yap, who's over there, actually found that when he transplanted my cells into mice with liver disease, they generated liver tumors. Okay? And that set me back because I thought, well, okay, one has to be very careful when you go into the area of stem cell therapy, because if these stem cells can be Jekyll or Hyde, right, and it gives rise to tumors, then you've got to be careful. And a colleague of mine, I don't call him a friend anymore, wrote this editorial in Cell Stem Cell. He actually put the question up in an editorial, liver stem cells, where art thou? Markers and two other laboratories looked for these stem cells in a functional way. They wanted to show that these cells actually transitioned from liver stem cell into hepatocyte. So they did tracing experiments. You can now mark transgenic mice genetically and follow the lineages. And they never found, when they marked the liver stem cell, they never found a hepatocyte that bore that mark. Correct. But when I looked at their experiments very, very carefully, they did not satisfy one caveat. And that is, you have got to stop the hepatocytes from proliferating. If the hepatocytes are active, they do the job. There's no need for liver stem cells. Okay? So if you trace, you'll find hepatocytes being traced to hepatocytes. You don't find liver stem cells being. So I wrote back to the journal and said, look, I challenge this. I've got this evidence and that evidence. But they said, if you're writing a letter, your challenge is far too long, shorten it. And I shortened it and went back to them and said, you have to make it shorter. I said, if I make it any shorter, I can't make my case, so forget it. Anyway, this was a huge problem for me. This came up as I was writing grants to get NH and MRC grants. So guess what my reviewers came back with, right? First of all, we're not sure whether these liver stem cells exist. And others say that if they exist, what function do they have anyway? Because the hepatocyte can give rise to hepatocytes. Why invoke liver stem cells? So this was a huge problem. I had about 3 or 4 goes with the NHMRC, and I eventually gave up this line of investigation. So I guess the lesson is, you can bang your head against the brick wall a few times, but it's hopeless when you're faced with this kind of editorial, because this paper was cited in my rebuttals. Okay, so I switch emphasis then from using these cells for cell therapy or trying to say they do great things. I switch to using them for drug discovery, and for drug discovery, I use these liver progenitor cells as a platform to screen for anti-liver cancer drugs. I had an advantage. I twisted the story around. I had good cells, and cells that you put into mice and they give rise to tumors, and we have transformed and non-transformed liver progenitor stem cells. We have these in our hands. We could test one, it doesn't give rise to tumors when you inject it into new mice, another one does. Okay? And what we have, which is unique, I think, in this field, is we've got the before and after. So we've got the good apple and we've got the rotten apple. Okay? So I thought that we could use this to advantage as a platform for screening. Second, we could also use it for biomarker discovery, because my collaboration with Martin Pera over in Melbourne, he made an antibody that could detect human liver progenitor stem cells, and it was a surface protein. And he showed in the pancreas that the pancreas, if it's cancerous, produces a protein that this monoclonal antibody reacts to. And he was able to detect some of the protein that he detects is shed from these cells into the serum. So he had actually taken out a patent to use this monoclonal antibody for what we call GCM5 from now on, as a way of detecting pancreatic cancer. So we know it's a protein that decorates the surface of the cells, and it is shed into the blood. So I thought, well, let's see whether this actually marks progenitor stem cells in humans under those different conditions. And indeed it does. So the drug discovery part, I will go through now and tell you about funding for that, right? Because Darius told me, you have to tell them how you get money. All right. So here, the funding actually came from the Busselton Jetty. They had already started fundraising, and they had a golf day. At the golf day, they raised $50,000, and they said, George, what about screening your liver stem cells with extracts from our sponges? So Lisa Shreve, who's the CEO in charge of the jetty, and Danelle Dowding, who's very interested in the science of the sponges, got in touch with me and Gavin, and that project has been launched. You can go to their website and you can see what we're doing there. There was also Chris Rickstein, a farmer down in Esperance. He passed away, no relatives, and his love was exploiting the flora and fauna that's unique to Western Australia, and he wanted us to do something about using these to find anti-cancer agents. That was his remit. So the biota pipeline was started up, and in fact, Chris was persuaded to do this, I think very much by Peter Klinken, who went down there, chatted with him, and mapped out the biota pipeline. So these were the funding opportunities that were present. I think it's also important, if you're going to go for funding, you have to have a team assembled. And what I've illustrated here is that I had the LPC lines, live cell monitoring, and my expertise in cell culture and liver cancer and liver. Gavin Flamenti had the license to collect through WA Agriculture and Fisheries, his contacts, so that he already had licenses to collect the stuff from the wild. And Gavin was an expert with extraction, purification, and identification of compounds. I also had a collaboration going with Scott Stewart. Scott, mass spec, he could identify what the compounds were, but more importantly, Scott was an expert in what they call, they have a method of looking at molecules and then modifying them to make them more or less effective. Right? They could see grouping, so they've got different eyes to us. And Scott brought in drug design, chemical modification, and in fact, the ability to synthesize from scratch some of the compounds, not all of them. So we also had to secure funding, we had to prove collaboration. So here are papers that I published with Stewart, and what that tells you that he had a thalidomide derivative that he modified to reduce its properties of causing problems with morphogenesis, but increasing its efficacy towards to be an anti-cancer drug. So this project is actually still ongoing. We don't have funding for it, but this tells, when I put this in, I'm saying I've already collaborated with the chemists and the people who are actually obtaining the material. So how do we get going? First of all, you have to have some preliminary data that says that this is a feasible project and that we've got some tantalizing data that we'd like to continue this work. So what happened here was, so in 2023, I had $15,000 left in my grant, my account, and I was pretty well close to closing my shop, because I was going to tell Heloisa, who was working for me, that we've got 15,000, and when you run that out, that's it. We're no longer open for business. Fortunately, the Cancer Council gave me a Career Achievement Award and gave me $20,000. I put that $20,000 towards this project, and the Busselton Jetty had a golf day and came up with $50,000. So we were able to pursue this project. But we're close to not doing it at all. So thanks Cancer Council and thanks Busselton Jetty. So the seed funding then generated preliminary data that allowed us to persuade bigger grantors to be interested. So this was a pilot project with that money that we got. Here are the sponges that have been collected by the WA Museum. And I didn't know anything about this, but they apparently dredged this area and came up with about, well, I got 80 sponges out of that, different types of sponge. And we then started working on that. This is a collaborative project between the museum and the Australian Institute of Marine Science. They were dredged and they collected 146, but 80 specimens were frozen at UWA, and we made extracts from this. So to cut a long story short, one sponge, and the IP people told me that if I reveal what that sponge is, I'm going to be in big trouble. So that's why that's redacted. But that sponge produced a very, very effective anti-cancer agent. So what I want to show you here is that this is the tumorigenic liver progenitor stem cell. In green is the vehicle. In blue is the approved drug, which we use sorafenib, which is what the clinicians use, and it's very effective. Kills the tumorigenic liver progenitor stem cell. The sponge extract completely wipes it out. But what I want you to focus on is over here. This is the non-tumorigenic liver progenitor cell. You can see the sponge extract doesn't affect it, whereas the approved drug does affect the normal cell. So here we have a proof of selectivity in this agent that we've extracted from the sponge. Okay? So whoopee, I thought, well, great, We finish off with that little piece of sponge. I said, let's get some more because we want to actually purify this compound. We want to fractionate it, and then we want to test it further. Unfortunately, that's all there was. I said, well, send somebody out and get some more. I said, nobody's going to dive a kilometer down, and they don't know where to go. Because we didn't GPS tag. They didn't GPS tag where the sponges were collected from. Okay, so this ends this project, in fact. We couldn't do any more with it. We ran out of material, and we ran out of sponge. I told them, why don't you send somebody down with a torch and dive down a kilometer and find it? But they said, no one's prepared to do that. Anyway, so that at least gave us some preliminary data that says that we can look at the 30 sponges from the Busselton jetty. Okay, so the biota funding now has kicked in, and this has provided us with 2 years of support and to do a wide screen. So the screen now will involve 226 sponges, including 30 from the jetty and the rest from the WA Museum collection. And they want to test sea cucumbers, some native plants of 500, and some plants from the indigenous owners in the Bardi territories. So this is on our plate for the next 2 years. Quite a lot of work to do all of this and screen them. So the idea is we screen first just the extract, and then if the extract is active, we'll fractionate, test the fractions, and the active fraction we purify and try and find the compound. So to summarize, orders from Darius about funding, the right fit of grantors and grantees. Make sure you're eligible and you're competing in a space where you have a good fit. Ensure the infrastructure is satisfied. The ethics and the permits are already are ongoing so that you can mount the project. Have the right team, right? I didn't put it in the ground and say, look, I think this would be a great thing if I can hire a chemist to do the job with me. I had the chemist there already. So if I walk in the room and say, look, I'd like $150,000 to hire a chemist to work on this project, the reviewers would say, who are you going to hire? How do we know you're going to be able to deliver, right? But I've got people on board. I publish with them. They're experts in their field. So I think that gives us an advantage. And also that our timing, when we got the small grants, the Biota grant round came, and we were really in a good place we're well placed to secure the Biota funding. So I'll give you a progress report on the drug discovery. We initiated this project actually in July 2025. So we've gone about a year, and we made pretty good progress. So here is the illustration again. Here are my cancerous liver stem cells, the bad guys, the rotten apple, and the non-cancerous liver stem cells, which are the good guys. And these ones give hepatocytes, these guys gives hepatoma. So we now use this as our platform for the screen. And here are the divers from the Busselton jetty. I told them to put George on that, but they didn't. Anyway, they collected 30 samples for us. ground them, and here they are. And Felicia, who's working on this project, she operates the robot to do all the pipetting, and she operates the microscope in an incubator that monitors the cells and puts little colors around them and counts the number of cells for us. And you can look on your phone and it tells you how many cells you got growing in the incubator. It's wonderful. So you know when to come in and subculture and start the 96 well plate experiment. So what we have now is all these extracts. These are all just Bustleton from the sponges. Look at the beautiful colors. This is our microscope in an incubator. It does all sorts of formats, and this is the output of the data. So you can see from this which cells are growing, which cells are not. And Felicia and Mike are the people at the coalface doing this work. And Darren Holland has joined the group. He's funded by Biomarker. He's a natural products expert. Darren is now actually doing the part of, we test the extract. He's actually taking this and he's using chromatography to purify, to separate these into fractions. And for each extract, gross extract, he's giving us 7 fractions, okay? And then we test them. So what are the results? Well, he has used mass spec and NMR, and he's identified novel compounds in some of them. So these compounds have not been associated with sponges before, and we've got new compounds. They have not in the literature yet. And we have fractions that are now highly effective. 6 of the extracts out of the 30, and I got very uncomfortable with this because a very high hit rate, 6 out of 30. But I'm told sponges actually put out very, very noxious agents. Why? Because they can't run away when they're... So they make something, they put it out into the seawater, but it has to be very, very effective because it gets highly diluted. Right? And you'll find, I was told by the divers, one pylon, usually one sponge occupies it and says, this is my pylon, the rest of you stay away. Okay, so they have agents that actually says, this is only good for me. And sea cucumbers is another thing. They put sea cucumbers into aquaria and everything else dies except the sea cucumber. So these guys are very, very territorial. And by virtue of that, they make quite incredible looking compounds that the synthesizers have a problem making. Okay, so with the drug discovery story, the next steps are now we have 6 fractions that are very effective on cancer cells. We are now testing them on the normal cells to assess selectivity. We will identify the compounds in the fraction and then we'll test for toxicity and selectivity. Scott Stewart's group will modify the compounds to enhance selectivity. And he can do that once we identify it. And I am interested in determining the mechanism of action of the hit compounds. What part of the cell cycle are they working? What part of the oncogenic pathway are they working on? That's something that I'm interested to learn and understand. We will expand our bioprospecting program by inclusion of sea cucumbers, native plants, and fungi. And while we've been doing this, we've heard from all sorts of people out there. Friends of the Tuart Forest got in touch with us and said, we go out into the Tuart Forest and we find all sorts of stuff. Can we collect them for you so that, well, I said, first of all, you better make sure you have a license to collect the stuff, right? So get in touch with Ag Department, Wildlife, etc., and make sure you can do that. But we'll happily include you. So that was going to happen. And of course, the Scientific Advisory Committee of Biota have got some very, very smart people on it. And we got somebody, Vicky Avery, from that organization. She was leading the AstraZeneca $20 million bioprospecting initiative. And I don't know why, but AstraZeneca, after a couple of years, said, we're pulling up stakes. There's $20 million worth of equipment here. You can have it, Griffith University. It's beautiful. So Vicky is running that facility. And we will probably, our hit compounds will send to Vicky to do tests across a wide range of different cancers. So that's what we'll end up doing. But she has informed us that my mouse cells are not human cells, and we need to test them on human liver cancer lines. Okay, so I'll switch gears now and go to biomarker discovery. And once again, I'll just go through with you. Funding, as I told you, I had a problem with my getting money from the NHMRC, because also, when you look at alcohol-induced hepatitis, fat-induced hepatitis, viral hepatitis, all these are lifestyle-imposed health problems, okay? And it is very difficult to get people to fund these, either from philanthropy or from organizations, because it's hard to raise money to support people who, in quotes, bring this on themselves, right? Nevertheless, with my GST-M5 monoclonal antibody, I could detect the stem cells in each of these. And I presented these results, and the GST-M5 monoclonal antibody does detect something in the sera of these people. So I had some preliminary data. However, when I went to cystic fibrosis, that's a cystic fibrotic liver, and I got funding, okay? So there is a difference. Go to the right place, and you have a sympathetic funder who's prepared to give you money to start you up. And in fact, Conquer Cystic Fibrosis gave us the money to start this whole thing up, all right? So we got funding. I also wish to, I'll go on with this, to getting the right team. So myself and Martin Pera, we had the monoclonal. He's given me the hybridoma, and we can identify and quantify human liver progenitor stem cells. Grant Ram and Peter Lewenden from the QIMR Berghofer have been working. I looked up, and he's got lots of NHMRC grants on cystic fibrotic and liver disease. And he has a biobank that is worth its weight, not in gold, but whatever is the most valuable element. He actually has liver biopsies from kids alongside sera that are matched. So we can get sections of the liver to look for our liver stem cells, and if they are there, we want to see whether the protein is shed into the blood. Okay, so that is a huge thing. Stephen Stick and Marcelo Lear are local. They are looking at kids' respiratory issues, and Marcello is the gastroenterologist at the Children's Hospital. Unfortunately, this access that we were told we will have access to the cystic fibrosis biobank and their clinical expertise, that's what they brought in terms of resource. But the biobank, my understanding is that there's a moratorium on the biobank due to some issues with its use, and that is still ongoing to try and resolve. When that's resolved, we'll be able to do more and extend our study. But they are the respiratory clinicians, scientists, and pediatric gastroenterologists. And Ingrid Lang was a respiratory scientist that first got me a bit interested in cystic fibrosis and liver disease. I spoke to her over cups of coffee, and I suddenly realized that Ingrid was in fact contributing more intellectual input than I was into the project. So I asked Ingrid if she would be a CIB on the project. It was only later that I learned that Ingrid actually is cystic fibrotic. She's had a double transplant. She's had a lung and a liver transplant. And she's actively researching, and she's a great source of, she brings the lived experience to the party. So once again, we have published with RAM several papers. So we've proven that we work together. So we published J. Hepatol, we published in Cell Genomics, and we've published American Journal of Pathology. So the first thing we did was, can we use the monoclonal antibody GST-M5 to find something in the blood of these patients? So here was the first experiments that we did. the seed funding actually came from the Royal Perth Hospital Research Foundation, in the sense that the executive and the board were good enough to allow me to convert my salary into an honorarium. I used that honorarium and put it into my research grants. So I am grateful to the Royal Perth Hospital Foundation for allowing me to do that, and I could then, if it wasn't there, once again, my lab would have closed, okay? What we did here was actually do a preliminary study, and this was done by Eloise, and I was about to tell Eloise that there's no money and you've got to go and find another job. But what she did was to assay a few samples that we got with people with fatty liver disease versus normals. Some are mildly affected, and we could see in their blood higher levels, and here in that triangle is actually a person who's got fatty liver disease and then progressed on to liver cancer. So one, we can measure it in the blood, right? And possibly the levels may reflect what is happening in their liver in terms of the population of liver proteinase cells. Okay, so with that information, we then went to our Conquer Cystic Fibrosis, and we said to them, we have the antibody, we have livers from kids with cystic fibrosis, some no liver disease, some with liver disease, various levels of liver disease, and the liver disease is quantified by the fibrotic score, F1, F2, F3, F4, or F0. So the mild F1s, moderate, you can see now my antibody is marking some cells, and in severe, the antibody is marking lots and lots of cells. Okay, so indeed, when we use this grant, we could identify that in the liver of cystic fibrotic kids who have a liver comorbidity, there are liver stem cells. So they are not unlike the alcoholic, they're not unlike the viral hepatitis patient, and they're not unlike the genetic hemochromatosis. So their livers are under enormous stress. We also later then got funding from Cystic Fibrosis WA and Telethon to support our efforts to assess the utility of the GCM5 monoclonal antibody as a biomarker. So from these conditions, we got blood and we got cystic fibrosis to fund the ELISA arm. All right, I'll just keep talking and wave my hands around. So the situation then is that we got funding to do an ELISA assay, and we said if we can do the ELISA assay, we build on to the Luminex assay, which is more sensitive and also more specific. And with our monoclonal antibody, we could make our own kits so that we're completely in control of what reagents we use. As a backup, and my philosophy of doing research is try and run plan A and plan B. If you can afford it, run them in parallel. Because if both of them work, you've got confirmatory data. If one of them doesn't work, maybe the other lane will do well. So that's what my philosophy is. Therefore, we scoured around and we found a company called Cloud Clone that actually makes ready-made kits to assay this GCM5 epitope. So I bought the kits and we ran commercial versus in-house, ELISAs and Luminex. Okay? What I'm about to tell you is that I'm going to bare my soul. The results that we got from using those kits and our kit is confounding. For a start, our kit didn't actually measure anything in the cystic fibrotic sera. The commercial kit did. So I concluded that maybe, I was going to show you the analogy of the canary. The canary is the liver progenitor cell in the liver. The feather is what's in the blood. But maybe not the entire feather is in the blood, right? So if we talk about the C terminus and the N terminus of the feather, maybe one end of it is in the blood and our antibody doesn't see it. So to cut a long story short, what we did was we pulled down the protein and did mass spec analysis on it. We found that our antibody actually reacted to the C terminus. It turns out that it's the N terminus that's in the blood. And that's what the polyclonal antibody, that Cloud clone, reluctantly they had to declare to us. And there was also a lot of problems. And I think you should take this as a warning that when you buy commercially produced reagents, they are not always what they seem, okay? Because the ELISA assay worked, and when we bought their Luminex kit, it didn't work. So we went back to them and said, what's the matter? They said, we have to tell you that the antibody that we use in the ELISA kit is different from the one in the Luminex kit. And they said, we cannot tell you why we changed them. But since you already spent many thousands of dollars on the kits, we will make you a Luminex kit that has the same antibody as the ELISA kit. And now both the ELISA kit and the Luminex kit worked. Okay, we're back. So that really is the story I was going to tell you. In fact, how are we going for time? All right. So I think I can go back and, can I go back? Yeah. So here is the puzzle. Like I just summarized, the assays of the CF children yielded confounding results. Our ELISA kit didn't work. We used a commercial ELISA kit that worked, but the Luminex assay kit didn't work. They are polyclonal antibodies, right? However, levels are variable, and in fact, we got the opposite result to what we expected. The F score, the fibrosis score, was inversely proportional to our biomarker. So it's really confounding. Some of the reasons for this is, as I told you, our antibody was seeing one end of the protein, and their antibody, which is polyclonal, saw the other side of it. So we did a pull down and mass spectrometry of serum, and that revealed that our GSTM5 antibody binds to the C terminus of the biomarker. And it's our guess that the feather gets chopped up and the C terminus is not in the serum. All right. And another thing, the commercial assay, we also, okay, so maybe I take a step back and tell you that I suspected that we would have to age age match this data with normal kids. Why? Because the progenitor cells, I believe, would be present in newborns, and it will progressively decline as the number of stem cells in the liver of these kids declines. And we approach Melbourne. There's a Happy Kids project there where they collect sera from our children, and I couldn't believe this. We had the ethics done in 2 months, and they sent us 380 samples. From year 1 to year 18, 5 females, 5 males, normal serum. We don't have the money to analyze all of them, but we've done selected ones, and we know that our biomarker declines with age. So that is another confounder. We have to age match our cystic fibrotic kids' levels. So at the moment, this is really bewildering us, and we are, I've lost, not changing. Okay, so that's the story. The N terminus is there in the blood, and the commercial kit can see that. Our kit, homemade kit, sees the C terminus. So we now purchase the peptide from China Peptides. They made it for us, and we're currently generating antibodies to the N terminus so that our homemade kit can mimic the commercial kit. So that's where that's at. But as I say, my policy is not working.
Recording in progress.
Okay, so again, while we're doing our work with our biomarker, we also use a proteomics approach. And mass spec is one way you can look at target proteins, but this came on the market. And if you're looking for biomarkers, this is a very interesting approach. SomaLogic actually makes fragments of DNA that binds to specific protein structures. They've got 11,000 aptamers which will bind a protein specifically. And then to quantify that amount of protein, they use PCR to actually, the amount of DNA that they bind is proportional to the amount of DNA they can assay by PCR. So with that, it can actually, we found 10,776 protein targets in our serum. We are 46 cystic fibrotic patients with mesh liver biopsies from F-scores from 0 to 4. And our aim was to assess the utility of SomaLogic's. They've already got a platform where they are identifying patients with liver fibrosis. So that is part of the commercial undertaking. And we also want to look at these 10,000 proteins and see whether we can come up with our own panel. And finally, that's what we wanted to do, develop a cystic fibrosis biomarker panel. And we also discovered a whole lot of proteins using this approach. So here is what are called volcano plots. It's not showing up very well, but that picture, that's a volcano. And here are the overexpressed genes, 399, and here are the underexpressed genes, 87, when you compare fibrosis 2 versus 3. When you compare fibrosis 3 versus 4, you also have upregulated genes and downregulated genes. There should be dots all over here, but that's not showing up that well. So we now have a huge panel of genes that we know are upregulated in the cystic fibrotic kids, and some genes that are downregulated. When we do pathway analysis, I haven't got the time to show you here, but it totally agrees with what we think is happening. These kids are making fewer hepatocytes and fewer cholangiocytes. That hallmark is being downregulated. Instead, they're making lots of progenitor cells, and the progenitor cells are not doing the right thing. So this begs the question, as the cystic fibrotic kids live longer, the big question I've got in mind is, will they develop liver cancer? Right? So that's something that I love to do with the clinicians at Charlie's, because when the kids are more than 18, they're handed over to Michael Wallace at Charlie's. So we do have a fantastic outcome. SomaLogic's liver fibrosis panel of 8 serum markers actually correlates extremely well with the fibrosis situation. Unfortunately, this proteome analysis costs over $1,000. The 8 proteins, if we approach SomaLogic, say they can reduce the cost from $1,000 to $500. So we may do this in future, and I'm interested in, if I can get the funding, we'll do the happy kids, the 360 samples from the happy kids, and put it into this system. But we have come up with 4 serum proteins of our own, and it is superior to the SomaLogic setup. Because what it shows you here, if you look at this graph, the distinction of our panel of 4 proteins, right? The area under the curve is 0.888. And it distinguishes the F2, F3 well from the F0 and F1, in contrast to the SomaLogic. So our next aim now is to take what we know about these biomarkers and put them into a mass spec, because the Proteomics International tells me that if I want to assay these 4 proteins in serum, they can do it for around $25 an assay. So this is the next step. That's what we'll do with. Our aim is to get funding, and we'll do the mass spec analysis. So I want to finish off with acknowledgement. The drug discovery, the liver cancer part, Felicia and Mike are the workers at the coalface. Gavin and Darren are doing the extractions, and we're grateful to the divers and Lisa and Danelle from Basel Jetty and the Biota Pipeline for funding this. Biomarker discovery with cystic fibrosis, Adam Passman and Augustine Skro are working on this. Stephen Stick, Marcello Leal, and Ingrid Lang from the kids are our collaborators, and Grant Ram and Peter Linden at QMI Berghofer are providing the very, very valuable samples. So I thank you for your attention, and I'm very happy to answer any questions.
Thank you so much, George, for sharing this exciting journey, and just remarkable, how much you can do over, a career of research. We're very glad to hear that you've chosen research as your place to go to, even though your mom didn't seem to be all that happy at the time. We have perhaps 2 or 3 quick questions. May I start with the last one? What made you think in the first place that those sponges may be a source? How on earth did you think about sponges down there?
Exactly.
A kilometer down?
I mean, that's the question I asked the people at the jetty, and they said sponges, when they are threatened, cannot run away, right? And because they can't run away, they make very, very noxious chemicals. And some of the chemicals that we have isolated from them are remarkable structure. 7 or 8 benzene rings and long chain fatty acids, and they can bind to all sorts of things. So it'll be interesting, I think, when we find the active ingredients, anything that kills the cancer cells, we should use these and find out by what mechanism. Because our assay at the moment tells us that some of the drugs that we've got actually just slows down the cell proliferation. Some are actually toxic and they kill the cell. So they are working through 2 different mechanisms.
Amazing. As you would know, a lot of the pharmacies or the drugs we use, snake venom, bark trees, and just remarkable where they come from. Yes. So, that's a great idea. Maybe we should collect samples wherever we go, a bit of bread. Any questions from the audience? Yes, please, Trevor.
Just a quick comment. I mean, looking at sponges is not new in itself. Back in the 80s, I think there was the AIMS Institute, Australian Institute of Marine Science over on the East Coast, and that's what they were doing, extracting sponges. But interestingly, when I was doing my PhD, there was a PhD student working with Emil Ghisalberti, who you probably know. In fact, Gavin is a student of Emil's. Yes. They discovered the smoke bush compound. He extracted dozens and dozens of sponges from the southwest coast. It would be useful for Gavin to look through Rob Capon's thesis, because you can't tell us what the name of that sponge is, but it may have been extracted already by Rob, and he actually pinpointed a lot of where he was collecting, and he isolated the pure compounds.
That sponge is like that was the best hit we had out of the 80 sponges. Yeah. One was not that selective, but somewhat selective. But all of them, they're gone. We squashed them up, extracted methanol, and then threw away the rest. But I thought, get some more. But I said, nobody's going to go down a kilometer to pick them up.
I mean, just following on quickly, Emil's passed away, but I know he kept everything. They may still be there. All of those extracts that Rob extracted may still be there.
That's interesting.
You should get Gavin to check.
Well, I knew Emil very well.
Yeah.
He married Anne Armstrong.
Before we dive deeper into the family, we'll try to grab that sample. So, look, for the interest of time, please join me in thanking George again for an amazing presentation. Thank you so much.
Thank you. Thank you very much.